Hello and welcome to our video on cellular respiration's glycolysis. This is one of 3 videos that are going to look into detail on respiration. Before we go further, I just want to start with this word glycolysis. We have the beginning starts with glyco, which when you hear the word glyco, you should think of sugar. Like, I would like some glyco in my coffee, please. Or can I have some glyco in my iced tea? So just think of glyco as referring to glucose, which is a sugar. And lysis, we know, means to break. So essentially, in this process, we are breaking apart sugar. Specifically, we're breaking apart glucose. Now, we understand when we studied hydrolysis that we used water to break apart different molecules. So kind of lysis is being used in the same context. So let's take a step back and look at our big overview of cellular respiration. We have glycolysis, which we are talking about today. And glycolysis takes place in a cytosol, like the cytoplasm, same thing. And it gives us 2 ATP. And we know that glycolysis is a process that doesn't require oxygen. Another super cool thing that glycolysis gives us is NADH, NAD. And NAD's whole job is to carry electrons to pretty much like the ATP moneymaker, right? The big factory, the electron transport chain and oxidative phosphorylation. Those are 2 processes that give us a ton of ATP. So we are going to just be taking a big look at this step 1. Our next video is going to be looking at the Krebs cycle in step 2. So this is when we, kind of check into the mitochondria and see what's going on there. And last but not least, we will be looking at electron transport chain, which also takes place in the mitochondria. So without further ado, let's get started. So glycolysis, where does it happen? Where we know it happens in the cytoplasm of the cell. And what happens? Well, effectively, we're taking glucose and we're breaking it down into 2 pyruvates or pyruvic acid. We also get a few other things from there. We make some ATP, we make some NAD, which is an electron carrier, which we will talk about. I like to think of glycolysis as making lemonade. So think about everything that needs to go into making lemonade and how that is helpful for you in your bank account. So let's take a look at the details. Here we have let me do a different color here. Here we have this is glucose. This should look really familiar to you. So this is glucose. It's a 6-carbon sugar, and it's often represented in a chain. Well, what I'm going to do to this chain is we're going to break it, and we are going to kind of like if you were taking off a necklace, we're going to lay it out straight, and we're just going to look at the carbon backbone because that's going to help us simplify, and we don't really need to worry about what's going on with all the hydrogens and oxygens. They're just kind of along for the ride. So we're going to start off with 6 carbon chains. See if I can count. 1, 2, 3, 4, 5, 6. Woohoo! College. So we have 6 carbon sugar, or yes, exactly, glucose. And we want to break that apart. So we are going to break this bond. And in order to break this bond, we need to, in comparing this to making lemonade, we need to spend a little money to get all of our materials, right? So we're spending a little money to get our lemons and the pitcher and the sugar and the cardboard boxes to set up our lemonade stand. So this takes an initial investment, and that initial investment is going to cost us 2 ATP. Okay? So our lemonade stand, you can say, oh, we've invested this much money. For glycolysis, we've invested this much ATP, but we're hoping to get more than that back. So if we take a closer look, this ATP breaking apart these carbon chains, we're going to get 2 special molecules that have 3 carbon chains. And they're a 3 carbon chain with a phosphate group attached to the outside of it. So here's our little phosphate group here, and our little phosphate group here. And this is essentially a 3 carbon chain that's been phosphorylated, and they are called, let me get my different color here, these are called PGAL. And PGAL stands for phosphoglyceraldehyde. You don't need to know that, but just know it's 3 carbons with a phosphate attached to it. Now, in order for us, our end product, we want to eventually get a little bit of ATP, we want to get some electron carriers, and we want that pyruvic acid. The pyruvic acid is going to be like the brick and mortar to building our ATP empire. So we go through several other processes, and these are all done by the use of mostly protein kinases. There's about 10 different proteins involved in 10 different chemical reactions to get us from our original glucose molecule to our final product, pyruvic acid. So we eventually end up with 2 pyruvates. Oops, that's a C, just so you know. C. Okay, so we eventually end up with 2. These are pyruvates. They're also called pyruvic acid. Those words are interchangeable. You can use either one. Now, in this process that protein kinases are used to break this down, we also get a few other things. I'm just going to draw it on one, but know that it happens to this one as well. So we are, in this process of breaking down, we actually get 2 ATP. Now we get 2 ATP from each of them, right? I'm going to go ahead and draw another one. So we get a total of 4 ATP, but we invested 2 ATP. So if we invest 2 ATP, we get 4 ATP. Now that's a pretty good return. If you invest $100 in your lemonade stand, this is the portion where we're starting to sell our lemonade, right? The payoff phase. And if we invest $100, this would be like we are getting $200, okay? So we invest 2 ATP, we receive 4 ATP total. Another thing that we get is this molecule called NADH. NADH. And NADH first started off as NAD+. And then during this process, we were able to add a hydrogen ion to this molecule. And it's basically, we're reducing this molecule. So we're adding a hydrogen and reducing this molecule. And now this molecule has high energy electron attached to it, and which is then going to be carted over to the electron transport chain. So that is glycolysis. In summary, we start off with 6 carbons, 6 carbon glucose. We have to put in 2 ATP in order to break this apart. Once we break this apart, we get 2 molecules of PGAL, which is phosphoglyceraldehyde. And those are essentially phosphorylized glyceraldehyde. And during a series of reactions, our PGALs then eventually end up as pyruvate, which is our ending molecule. And in that process, we make a total of 4 ATP. We reduce 2 NAD+ molecules to become NADH molecules that will eventually go to the electron transport chain. Ta-da! There it is, glycolysis. So what I would like you to do, on the left-hand side of the page, you're going to create a magic box of glycolysis. The really important thing to remember about this process is what is invested in terms of what did it cost to make our lemonade stand, and what did we end up with at the end of our big lemonade stand sale. So you're going to have, you can use anything for your box. It can be a picture of your cat. It can be a heart, it can be a candy wrapper, it could be anything that you want to use to symbolize your box, because basically this is going to be the process, which for all purposes sake, is essentially magic. I mean, when you think about it, it's 10 different protein kinases used to make 10 different reactions, which break down glucose into pyruvic acid. So we want to be concerned with what is going into this, and eventually what will be coming out. So investment and payoff. So that is your assignment on the left-hand side page of these notes. And I hope you enjoyed our video on glycolysis.